Okay, so let's look at protein synthesis. Protein synthesis has these two major processes, transcription and translation. Transcription is how we make the mRNA. And we're going to talk about the three different types of RNA in a little bit, but the mRNA is basically going to carry the instructions for how to make the protein. But transcription is how we make that mRNA from a DNA template, and that happens in the nucleus. And translation is how we actually make the protein. And we make the protein using the instructions on the mRNA, and we use rRNA and tRNA to help us do that. And that all happens in the cytoplasm, either on a free-floating enzyme or free-floating ribosome or one attached to the RER. To talk about the RNAs, we need to understand their differences. So one difference is in the sugar. The sugar in RNA is different than the sugar in DNA. And the only difference is it has its added oxygen. So in DNA you have deoxyribose, and in RNA you just have ribose. DNA is double-stranded, while RNA is single-stranded. And that's not true all the time, but for the most part and for our purposes, we're going to say that's the truth. And then the bases in DNA, there's one difference. Thymine is found in DNA, but in RNA, thymine is replaced with uracil. So it's adenine, cytosine, guanine, and thymine in DNA, and adenine, cytosine, guanine, and uracil in RNA. The RNA, so in RNA, adenine is complementary to uracil. So these two are complementary. Okay, three types of RNA. We've got messenger RNA. And messenger RNA is mRNA, and that's made from a DNA template in the nucleus. It has codons, and codons are just three nucleotide long bases of DNA. So the codons are just three of these. So 1, 2, 3. And those code for a certain amino acid. Then there's rRNA, which is ribosomal RNA. That's the RNA that's part of the ribosome. Remember way back when we were talking about cell organelles, we talked about ribosomes had two subunits. One of those subunits is the RNA part, and the other part is protein. And the ribosomal RNA, the ribosome, helps to hold mRNA in place so that tRNA can, quote unquote, read the mRNA. And then we have tRNA, which is transfer RNA, and that has an anticodon that will be complementary to a codon that's on mRNA, and it also carries a specific amino acid. Okay, transcription takes place in the nucleus, and it starts with the DNA being unwound and the strands separated. Then RNA polymerase reads the template strand, so the strand that's going to code for the protein, and adds complementary bases together to make the immature mRNA. It's called immature mRNA because it has introns in it. And the introns are just non-coding sections of the RNA, things that aren't going to actually code for the protein. So the introns need to be removed, and what's left over are exons. The exons are spliced together. The exons are the piece of the RNA that are actually going to code for the protein. And then the mature mRNA leaves the nucleus through the nuclear pores. So here's an example of transcription where we have a DNA template. And the DNA template's here in black, and it's CGTCT. You can read that. And first thing that happens is we make the immature mRNA. Remember, that's made by the enzyme RNA polymerase. So on the template where we have a C, the mRNA will have a G. Another G, a C, a T, an A C, a G, T, and A. And then here we go, we're going to switch because we have this A, and the mRNA is going to be made of a U, because remember, uracil replaces thymine in RNA. And for our purposes, and I'm making this up, we're going to say that all UGG sections are the introns. So all the UGG. So there's a UGG, and that's it. So that's our only intron. So that's going to be removed. We're going to take that out. And what's left over are the exons, and those two will get spliced together to make a mature mRNA molecule, GCA, GAC, CUA, AG. Okay, on to translation. Translation is how we're actually going to make the protein. So the mRNA, which is now in the nucleus, or which is now in the cytoplasm, is sandwiched between the two ribosomal subunits. When the start codon, and the start codon is AUG, is in the reading window, the tRNA that has that complementary anticodon, so what's complementary to AUG? It would be this, UAC. So that's the anticodon that's on the tRNA, and that's going to attach to the start codon. It doesn't really attach, but it makes a relationship. And then the first amino acid, which is attached to that tRNA, is methionine. So methionine is the first amino acid. There we go. The mRNA shifts to move the next codon into the reading window. So it's going to shift over. So the next 3 base long piece of mRNA will move into the reading window. And the tRNA that has that complementary codon will attach to the mRNA. The 2 amino acids that are now next to each other will form a peptide bond. And the first tRNA, once that peptide bond is formed, that first tRNA that had the methionine will now leave, and the process will continue until we get to a stop codon. And there's 3 stop codons. Okay. And this is just pictures showing that. So here we have our 2 ribosomal subunits. So here's one subunit, here's the other. This right here is our mRNA that's being fed into the ribosome. And here's the reading window. So there's the reading window. And here's the protein that's being made. All these colored balls here are the amino acids that are being peptide bonded together to form the protein. So if this was the first one, that must be methionine. Okay, good. And then here are the tRNAs. Notice that each tRNA has a, I'm just going to erase this for a second. There we go. On this tRNA, we have a anticodon, which it's hard to see here, but that's 3 bases long. That's going to be complementary to a codon. So this anticodon here is going to be complementary to those 3 bases. And that will carry its amino acid down. So there's the tRNAs, the rRNAs, and the mRNAs all working together. Okay, and then this is an example of the whole thing happening. So we'll start here with our DNA, and there's our DNA strand. We're going to make our immature mRNA, and our immature mRNA is complementary. So the G goes with the C. Now those are not bonds. I'm just highlighting that. Here, that G with that C, that G with that C, that G with that C, A with a T, U with an A, G with a C, A with a T, U with an A, so on and so forth. We're just matching up complementary bases. For this, our purposes for this one, we're saying that all the GGs are the introns. So here's some introns, GG. Here's some more introns, GG. So those are going to come out. And then one more set of introns. Here we go. That GG is going to be gone too. Okay, those introns removed. We have our mature mRNA here. And this mature mRNA is going to be able to leave the nucleus. There we go. Can leave the nucleus, and where these were all spliced together. Now, once inside the cytoplasm, it finds a ribosome, and the ribosome sandwiches the two subunits in there. And once AUG shows up in the reading window, then we have a start codon, and that forms methionine. Then we have AUA, isoleucine. This is all on, these codes are on page 338 in your book. You do not have to memorize the codes. You'll be given a chart with these codes. CGU and AAU, and then CGU's arginine, and they make a peptide bond, and then AAU's asparagine, and that's make a peptide bond. And then once we get to UAA, that's one of 3 stop codons. So UAA tells the, once that's in the reading window, that tells the ribosome that we're finished making this protein. Okay, so our protein is methionine, isoleucine, arginine, and asparagine. The code, remember there's 20 amino acids in nature, but there's 64 different codes that we could have. And so those 64 different codes means that some amino acids, actually most amino acids, have more than one codon for itself. And that's what makes our genetic code redundant. And you can see that on page 338. The only one that doesn't have more than one is the start codon, the amino acid methionine. That's only AUG.